Everyone, welcome to Mella's vlog. I have a really interesting article I want to read to you, actually two. So the first one I found on Newsbreak, and it's by Bible Insight. Now, it's not an article of faith, but they are referring to the biblical prophecies in this way. Scientists warn disease X is coming, echoing Jesus' warning of pestilences in Luke 21. This is a super short article, and then I want to go to another article on this disease X. Many people hoped the recent pandemic was once in a lifetime anomaly, but top epidemiologists in the World Health Organization are actively preparing for what is called disease X, and a future unknown pathogen they believe is inevitable and potentially more severe. Okay, we need to read more to really understand what's going on here. This aligns frighteningly well with Jesus' words in Luke 21:11. He didn't warn of a single sickness, he warned of pestilences, plural, appearing in various places as a sign of the end. We are living in a unique era where biology, global travel, ancient prophecy are converging. The frequency of these events is increasing. I mean, we see this. This isn't because we have internet and we can see more. It's because there is, in fact, more occurring quickly. The frequency of these events is increasing, much like birth pains described in Matthew 24. The contractions are getting closer. This isn't meant to scare you, but a wake-up call. What they mean by that is the more severity, this is what I believe they mean, the severity of occurrence, the frequency of occurrences is occurring more. But then the severity of each frequency of whatever it is that's causing illness is also occurring more. Okay, so that's basically that particular article, I thought. And then they have questions and answers at the end. Now I want to go to another article. So I was curious to learn more about disease X. What is this thing? And I found an article that was written in 2025. It's by The Scientist. It's .com, thescientist.com. And again, I don't have an author name. It's just by The Scientist, the website. So we're going to read it together. It's a little bit more to it, and just kind of do a reaction thing. I would love to know your reaction on this. So as we know, COVID took the world by surprise in 2020, being declared a pandemic in March of that year. While it was the most recent emerging infectious disease, experts anticipate that it won't be the last. And you and I knew that. We all talked about that. Co-workers talked about it. People talked about it. That if this occurred now, of course it could happen again. Unfortunately. So, well before the pandemic, scientists had been prepping for an emergence of an unforeseen pandemic, a disease X, also called pathogen X, in 2018. So I guess that's when they were expecting it. Who first introduced the term disease X. Okay. So pathogen X could be anything, including pathogen that we've already seen, and it just mutates in such a way that we had not thought about or anticipated. Said Karujic, I'm going to spell this because I'm not pronouncing it correctly, Kariuj, and Nienga, Nienga, is a virologist at the Washington State University. explores emerging and re-emerging infectious diseases. Something probably like the measles. That people can still get that. I mean, the measles, right? Chickenpox. All of these terms. Um, so it could be emerging or re-emerging, or it could be something that they've never seen before. Now, I'm going to say something I do know from studying chemistry and biology. The scary thing about these germs, or bacteria, or whatever they may be, they are alive. They're live. They're living. They're organisms, living organisms. And many times they will find a way to survive. So that's what they're talking about, is that as we go further and we learn to get rid of this one, we learn to get rid of that one, then, another emerges, even changing hosts sometimes, which is really scary. So, known pathogens offer clues for the disease X. That would make sense. Working full-time in Kenya as part of a non-government organization, WSU Global Health Kenya, which promotes public health and related research. Njenga studies how viruses that cause diseases like Ebola, Rift Valley fever, which I never heard of that one, Middle East respiratory syndrome, MERS. We've heard of that, and SARS. They're transmitted and maintained in the environment. Okay, basically. So having worked in the field for several decades, Njenga has seen the state of emerging infectious diseases. More recently, I've seen more emerging pathogens than we have ever seen before. So that's a clue that they could decide to assimilate, is what I'm saying. That's what I'm saying. One of the ways that scientists like Njenga tackle the question of pandemic preparedness is studying known pathogens. These familiar microbials threats have the potential to mutate, becoming more transmissible or acquiring new features that increase their severity. So with an organism, because it's so teeny tiny, the length of time that it can mutate or change may be very short. Not like humans where, it may take generations for something, maybe an appendage or something, to change in us. They can also serve as models for future similar but new pathogens. Njenga's group studies transmission and disease dynamics of MERS, MERS, coronavirus that emerged in 2012. Researchers found that camels served as the main reservoir for this particular host, with human transmission predominantly only occurring in hospital settings. That's what I was just going to say. MERS is the weird and very sad thing about MERS, MERS, and I've had family members contract it and also their lives taken with it. This is the thing, though. You go in with something, let's say critical, whatever it is. Maybe you've had a heart attack, maybe you have a pneumonia, whatever it is, some medical something. MERS is living on the surfaces of the hospital. And you're clean, and even if you're cleaning your loved one's room and you're cleaning and cleaning, still maybe the tray that comes in might have something of MERS, and the person who's already ill contracts this. It's very, very, very serious. So anyway, yeah, it says it lives in hospital settings. However, MERS infections in the Middle East and Asia have a lethal rate of 35%. In contrast, the strand circulating in Africa has not caused severe diseases to date in humans. Despite the fact that the virus is abundantly present in domestic camels. It's funny that they're talking about camels and then talking about hospital settings. So it just shows you how diseases and viruses and contagions can travel or be in one place and another very different place. The rare cases reported in Africa have been asymptomatic individuals. But because, hi, Umi. But because previous studies demonstrated evolution in other MERS strains, Njenga's team actively surveils the virus's genome to track possible genomic changes that may enhance the camel to human or human to human transmission. So he's studying the genes. Apparently, that's the genomic changes. Okay, so could this virus be changing genetically? And one of these days, are we going to have another pandemic too, kind of disease from this particular virus? Okay, so they're asking the questions, they're trying to prepare. These are the things we need to do to be ready and to be stronger and to know what is going on in our environment. And yes, we have our microbe bacterial environment, we have our ecosystem, we have our wildlife, we have us, you know what I mean? We have all these levels of environment. So Njenga explores the factors behind varying disease severity and survival in patients following Ebola, which is just a horrible infection. Although this virus is unlikely to reach pandemic levels, Njenga said understanding these different immune responses can provide insights into new therapies. We go in there and we ask, what can we learn from these to be able to respond better and be better prepared? That makes total sense. Thomas Giesbert, a virologist, also and he's at the University of Texas Medical Branch of Galveston, asked similar questions in his research involving emerging infectious diseases. Giesbert attributed his interest in some of the world's most dangerous known viruses to his curiosity. I just always thought it was intriguing to work with viruses and that that were deadly and try to come up with ways to stop them. So let me just, in this little box, they have a notation. Preparing for disease X takes a multi-pronged approach. So before I read this, so we really do not know what disease X is. They do not know. The scientists and experts do not know, but they do know that there is a possibility, and of the just knowing how microorganisms work and how they seek hosts and how they have the ability to mutate and different characteristics of these bacteria infections based on their knowledge and based on maybe some knowledge they still don't know. They are trying to study this and help us be ready. As humans continue to alter their landscapes and face more wildlife interactions, the risks of new infectious diseases emerging increase. I love the way they said that, alter the landscape. Yeah, the more acres and acres and acres of fields and woods that are gone, those are places that the bees and the insects, the rodents and the wildlife and the birds may all gravitate towards, and or be in, and be defensive, and or like we were talking over on Vermont Sun and Shade, my other side channel. If it's an owl, they may have a nest somewhere in that vicinity. They may go right for your head. And that's very serious because you may not survive something like that. So we talk about that. But to prepare for the unknown disease X, researchers are studying the composition of pathogens circulating in the wildlife. Monitor, yeah, because they typically are widespread. The closer they get together, the more they're going to have contagions between them. And the less room they have for the ones who are infected, which usually will walk off away from their group to be ill, they're going to be in closer than ever because they have nowhere to go. So for the wildlife, monitoring changes in the activities and environments that increase the risk of pathogens spill over into humans, and developing therapeutic interventions that could potentially rapidly be deployed. So the other thing I'd like to add to this, so they go on in this article, and I'm going to read a little bit here because they have a few more points here. One thing I want to add also is here in Vermont we have over 4,500 people who are homeless. Homelessness inevitably wears people down. It's not just mental and psychological and emotional, which is huge, but it wears you down physically. You can't get adequate rest at all. You don't have your downtime. You don't have space. You don't have comfort. You don't have consistently adequate food. And above all, you don't have consistent hygiene. This is a given. This is a fact. Even if you're in a shelter, you do not, because you're sharing it. Okay. Traditionally, research, and I'm just saying all of that because all of those are factors to contributing to people getting ill, contracting diseases easier. So traditionally, researchers identified new pathogens after they crossed over and caused disease in humans or animals. One plan to get ahead of these diseases, emerging diseases, is to better understand the microbes that are already present in nature, but characterizing these pathogen burdens can be complicated because of the physical constraints in collecting samples. Advances in diagnostic technologies and novel methodologies are expanding researchers' abilities to do this. To overcome limitations in simple collections from larger animals, scientists developed non-invasive techniques to use saliva from discarded food to detect known and possibly unknown pathogens using PCR. That's good that they developed that, because what they were doing is taking an incredible amount of time, a lot of money with the sedation meds, and putting these animals in the equipment, and then a lot of people paying all of them to put the animal down to get a DNA swab. So this is much better that they're getting that from something discarded that they've eaten. Meanwhile, improvements in multiplex assays expand the use of serology for large and small animals. Okay, factors like climate change, altered land use, and human migration are changing what animals people encounter, but the impact of these changes remains unclear. Well, I can tell you on a small scale anyway, but still, in Vermont, there's been an increase of bear to human contact and either or close vicinities, and there's been an increase in owl to human attacks and or flyovers. So those are two that I know of. So scientists are using the models in household and wildlife surveys to study how changes in the environment and behaviors are affecting what animals are present in the ecosystem and how humans are interacting with them. They can use the data to determine what practices pose the greatest risk to disease exposure. So I do know, excuse me, I need to take a sip of this. When you get, if a person gets attacked by an owl, the Italians, because I looked this up, but they're not poisonous or venomous, and that's what I was afraid of. But they might as well be, because guess what? The bacteria that is in the Italians or on them, once they get into, and they're going into your head, once they get into your bloodstream, they very, very quickly, as I understand it, cause infection or can. So you figure owls go for more rodents, chipmunks, rabbits, things like that. So yeah, there's going to be incredible amounts of bacteria already set in on their Italians, and that gets into your bloodstream. And you get a very quick, you have to go to a doctor immediately if you are attacked. Even if you don't think the scratch is anything at all, go get it checked and have them keep an eye on it because they know what they're looking for. Okay, so scientists don't know what exact virus may be the next to cause an epidemic or pandemic. However, they are developing vaccines and therapies like antivirals, monoclonal, that's what it says, monoclonal antibodies against highly shared elements of known pathogens. Researchers can have platforms ready to quickly design interventions. So that's some really good news there. The interest-led Ngenga to studying Ebola in the early 2000s. In 2005, Giesbert and his colleague Heinz Feldman developed a viral vector Ebola vaccine that fully protected monkeys. Oh wait, the interest led him. I thought they were talking about Nagenta. Let's see here. They're not clear. They skip from one. Or maybe they're talking, I'm sorry, about Giesbert. But that's the sentence. The sentence is, the interest led him to studying Ebola in the 2000s. In 2005, Giesbert and his colleague Heinz Feldman developed a viral vector Ebola vaccine that fully protected monkeys. However, the pathogens like Ebola, which cause sporadic outbreaks but don't reach the level of pandemic, Giesbert said that getting funding and commercial support to advance these therapies was difficult. This is like, he says, this is so frustrating for me for my whole career. So you would think there's always an ongoing sector of the branch of whatever it is in every single country around the world to be studying these potential pandemics or epidemics. Okay. The vaccine just sat there for years and years and years, and we knew it was good. Sadly, it took the Ebola epidemic in West Africa, and we remember that in 2014-15. I can't believe it was that far back now. That's over 10 years. I feel like it was just yesterday. I do. To get that thing across the finish line for somebody to show interest. Okay, so they're talking about funding. Okay, and the last part of this article is kind of interesting here. This is quite long, but it's talking, we're just going to touch upon it. Follow the food. New pathogen discovery approaches aid. Disease X prep, so preparing. Scientists may not be able to predict when a spillover will occur or which pathogen may become a disease X, but having a better grasp of a potential candidate can help inform researchers of what to expect and what to prepare for. Tierra Smiley Evans, an infectious disease epidemiologist at the University of California, Berkeley, studies the viruses that circulate in the wildlife, in particular the mountain gorillas and bats. She is also chief veterinary and scientific officer for Gorilla Doctors, a nonprofit organization for gorillas. Here's a picture of the gorillas that she's the head of the organization. And so what they're saying is they're going to keep an eye on the wildlife here. As a wildlife veterinarian, conservationists, especially endangered primates of the mountain gorillas, is important to Smiley Evans. Non-human primates are a really important part of the ecosystem. She said, explaining that these animals are susceptible to same infectious as humans, posing a risk for outbreaks that occur in one species and shared with another. Although the primates are not reservoirs of viruses, they can act as bridges between the wildlife that harbor the pathogens and humans. So in other words, you know, they're not, that they can give us, we can kind of get heads up from whatever it is they're contracting. We can get a heads up, oh my, this is what's going on here in the mountain gorillas or the mountain bats, or, you know, what's ever going on. Because wasn't COVID like contracted like from a bats, right? Anyway, it's really important to study these viruses at this particular interface where humans are moving closer into wildlife habitats that they haven't contacted before. That's where we potentially do the most, that's where we could potentially do the most benefit, because that's where we can catch these things, yes, before they spill over. So then she talks about here, yeah, trying to get some of the data from, or samples, collections from the species, from endangered species like the mountain. So I guess that mountain gorilla is an endangered species. For non-invasive testing, Smiley Evans and her team developed a method where she also uses like the saliva from discarded material. Okay. So, I mean, that's total common sense. I don't know why they weren't doing that like for years, but I'm glad they finally thought of it, because it's the most common basic way to do it, right? Anyway, so she says by looking at the conserved regions across the viral genome, the team can use PCR to detect both known and possibly novel viruses in these animals. With that being said, detecting a PCR positive sample in a non-human primate is like looking for a needle in a haystack. This is because most of these animals are healthy. And they're healthy because they're removed from habitats where there's this cross-contamination going on. You know, they're in environments that haven't been invaded or ripped up, or, you know, all of that. So to overcome this obstacle, her team is exploring using this method to detect antibodies in animal saliva. So they have to really work to find if anything is going on. So she goes on about that, and then it goes into how human activities could drive disease X into an outbreak. Human activity, not gorilla. not bat, not wildlife, but human activity. So we'll read this last part of it because sounds interesting. Beyond tracking the pathogens, researchers are interested in how people encounter pathogens and what actions may increase these risks. And this is by Chris Murray, is an ecologist at the London School of Hygiene and Tropical Medicine, studies this. So according to him, one lesson that became obvious following the pandemic in 2020 was the scientists aren't prepared enough to handle the next large outbreak. We simply don't know enough about the origins of these pathogens. We don't know enough about the ecology of the way that they move from their host to the human population. And you know, it's funny when you read that, because your common sense tells you, it's almost, to me, it tells me it's a step ladder. Like if, for example, let's say there's something in in a dog. Okay, let's say a dog somehow had contact with a fox, you know, a rabbit, something out in the woods. So it comes back, it's on the dog. The dog is in the house, so it gets onto the surface. So you think if this bacteria, organism, or whatever it is, it's alive, let's say, can live in the oxygen of the dog breathing. The dog breathes on you, or it gets on the surface and then on you. So I kind of think it like that as a step ladder, where it's just kind of hopping. But I think they're talking a little bit more in depth about how does it even survive from its host to another host and in between. I think that's what they're looking at. Let's see. So human activities influence the risk of spillovers by bringing people closer to wildlife in their habitats and increasing their exposure to novel pathogens. For example, Murray's group previously demonstrated how land use changes. Yeah, that's what I was talking about earlier, such as deforestation for agriculture or building apartments, increased malaria risk in sub-Saharan Africa. Additionally, they showed that these disturbances shifted the diversity in the bat species, which led to an increase in viral abundance in remaining animals. This is what I'm saying. When you upset the very land and environment and ecosystem that these animals and insects are dependent on, you're shifting them, you're moving them, you're making them move. It's very concerning to me, because I look at some of the things that have gone up and say that didn't need to go up. I mean, you have a perfectly, absolutely, you have acres and acres and acres of buildings downtown that are just dilapidating right there. Knock them down, clean it up, and use that area. Don't dig up any more wildlife areas. So, however, this is a bit of a gap. There's a bit of a gap in this particular component around human-animal contact, Murray said. Currently, most surveillance identifies a pathogen in an animal or a human. The bit that's missing in all of that for us is that very central process of human-animal contact, and whether that itself is predictive of the human infection patterns that we're seeing. Well, like I said, if it's in the animal and suddenly this human gets it, it's because somehow it survived the intermission, you know, the gap between being in the air or on the surface and then going to the human. To address this, Murray's group is interviewing households in Central River region, the Gambia, about their animal contact and collecting blood samples from humans and domestic and wild animals to compare pathogen burdens and exposures. Our plan is really to map the sharing network of pathogens and then link it to human animal contact patterns and look at the role of land use in shaping that sort of dynamic network. You know, I'm not a scientist, but I would think, yes, your environment, whether you're in the north, south, east, west, north, south, east, and west, even in just in the United States, for example, would affect DNA makeup, wouldn't it? Your genetic makeup to some degree, you know, for setting you up to be predisposed to something, or if you're setting you up against predisposition, because guess what? I could go to maybe another state and maybe I get bit by a bug or something, and the people there like get bit by the bug and they don't have a severe reaction because it built up immunity over generations. That's a very real thing. So you know, if you're exposed to a virus or bacteria that is so overt and, you know, severely attacks your system, certainly that can happen. For these studies, they are partnered with colleagues in Nigeria conducting similar investigations. Working with the anthropologists, the researchers also hope to be able to compare how cultural differences, I was just talking about that, cultural differences influence exposure risk, understanding how these contacts and activities that lead them to influence pathogen risk can help inform scientists and communities to better prepare. So the article goes on a little bit more and it talks here, which is also very interesting. I know we're at 36 minutes now. We want to maybe take 10 more minutes for this. This part of the article is talking about climate change shifts. I don't even need to read this before I say to you, the warmer the climate gets in certain parts of the world, the chances of epidemic or pandemic increases. It's got to. How can it not? Because that's where bacteria and viruses will thrive, is in the warmth. So let's see what it says. Another factor in shifting risk of emergent pathogens and disease X is the role of the climate change. There's no question that climate change is having an impact on the spread of infectious diseases and populations of wildlife where they can live and where they can move. She explained that 15 years ago, her team didn't need mosquito nets or anti-malaria medications. I disagree with that because it was over 20 years ago. I know that we had it was a person in town, not this town, it was another town in Vermont who took regular trips to Africa. One trip, she didn't have her malaria vaccine and she got bit and she did not make it. That was well over 15 years ago. They were giving the malaria vaccines. This lady is saying they didn't need it. They should have been using it, yes. She goes, now, but of course now it's strikingly different. Yes, it would be worse. To study these changes, her group traps mosquitoes across the research fields and throughout the seasons and samples their virus, assessing both numbers of mosquitoes and the variety of species. Meanwhile, in another project, the team compares serum from wildlife collected decades ago to that of the animals of the same geographic regions today to see how the virus profiles have shifted. It's going to be interesting to see what's happening as the climate changes. So what she's saying is that, yes, we've already seen a change, but she's not giving really the specific details, except for, yes, there's an increase. However, climate change induced migration shifts in mosquitoes and other animals are already changing disease risk. The mosquitoes are such a problem because they're like little flying needles filled with, yeah, blood. You know what I mean? And they go to person. They are such a, oh, do not like them. Murray's team and used a modeling to explore climate change driven weather alterations, increased genetic diversity and infection dynamics of the fungal pathogen in amphibians, contributed cholera outbreaks, and influenced the development of mosquitoes in different geographic regions. Just saying that they study those. They're not giving us the detail of the results here, except that that suggests that if you might have the right environment conditions and climate, now we have the virus everywhere, and you could get an explosive outbreak. That's the conclusion they're coming to with the climate change, is saying that the warmer it is, the transmission is more likely. That's what they're saying. Pathogen prototypes. Okay, so this is a study profiling wildlife pathogens and identifying risky behaviors are two arms of the three-pronged approach of preparedness. Even with all the knowledge on virus diversity and spillover risks, outbreaks will still happen. Yeah, that's true. But it may not happen on such a severe a level. Okay, so basically, that's a lot of the article. And they talk a little bit here on defense against the disease X. Are we there yet? Are we better prepared? Njenga mused. The answer is yes, with a small no. The speed at which SARS-CoV-2 was sequenced and made available, followed by the rapid development of vaccines, demonstrated the capacity of the research community to respond quickly. So that's good information. These advancements reinvigorated interest among funding agencies. Basically, this article, they're really talking about they need funding to continue their studies. And that's basically it. So it's a highly detailed article. I would say there's no argument about it. Is there a debate here? But so again, it's by thescientist.com, and the name of the article, if you want to study it and read it more, is Preparing for Disease X in a Changing World. And so it's very interesting. I will be back on here with a lot more things from sites like this and other sites, and just as our world is changing very quickly, and it seems on all levels. So with that, you have an absolutely beautiful day. Be safe and stay warm.